Pesticides are extremely important in the agrochemical sector, and their global production is growing by 11% annually. As of 2019, Ukraine ranks 6th place in the world in terms of pesticide use. Following primary effect, the pesticides residuals, which remain in the environment, are able to exert long-term harmful effects for physiological and biochemical systems of non-target animals. Nevertheless, promotion of green growth strategies and action plan for sustainable use of pesticides that contributes to further risk reduction all over the world. Studies conducted by US Environmental Protection Agency showed the presence of micromolar amounts of Roundup in about 30% of 154 analyzed river water samples. While neurotoxic effect of organophosphate pesticides is well studied in a wide range of organisms, the information about their effects on the health status in general are quite limited, particularly on the molecular level, as very appropriate for early warning analysis. Therefore, the aim of our work was to investigate the response of zebrafish to the effects of widely used organophosphate pesticides, namely Roundup and chlorpyrifos. We also have tried Chlorella as a putative bioremediative tool to reduce pesticides toxicity for non-targets. Toxicity of pesticides was evaluated on the adult zebrafish as a conventional biological model for mechanistic and toxicological studies. Experimental fish were exposed to organophosphates, namely Roundup, chlorpyrifos, and their mixture. We have used 15 microgram per liter Roundup and 0.1 microgram per liter chlorpyrifos. We treated animals for 14 days. Being experienced in multi-marker approach, we have carried out the comprehensive study based on evaluation of plethora of antioxidants, products of oxidative lesions, and signs of cytotoxicity presented on this slide. As you can see, total antioxidant capacity, catalase activity, and glutathione concentration were significantly decreased after exposure by organophosphate pesticides when compared with the corresponding control. In the same time, studied chemicals provoked an increase in lipid peroxidation, protein carbonylation level, and reactive oxygen species overflow, with the most prominent changes found in the Roundup group. No significant differences were shown in fish responses to individual and combined action of pesticides. Organophosphates switched off detoxification system in zebrafish liver, exploded by glutathione transferase activity and carboxyl esterase activity, and significantly altered phosphorylation process. In this case, more prominent changes were disclosed after complex action of two studied pesticides. Depression of antioxidants and detoxification process helped forward development of cytotoxicity. In particular, Roundup and chlorpyrifos caused an increase in DNA fragmentation and caspase reactivity, as well as methyl glyoxide, but only after chlorpyrifos treatment. As we have shown, Roundup and chlorpyrifos affect not only steroidal hormones pathways, but also hypothalamus, pituitary thyroid axis, and reduced acetylcholine esterase activity reflected neurological disorders, as was expected. The cytotoxic signs after combined action of pesticides were different from individual ones. It is highly likely that there was antagonistic relation while combined action. Overall, the classification and regression tree analysis supports an important role of the triiodothyronine in the response and adaptation of animals to deteriorated effects of pesticides, even in low environmental realistic concentration, what is very important. TBA-reactive substances and caspase reactivity also pertain to the most important parameters discriminating the experimental groups. Fish had affected by organophosphorus containing pesticides reacted similarly. Danio rerio treated by the combination of two pesticides were alike in the response to individual Roundup action. Given the present findings, we can conclude that Roundup and chlorpyrifos, even in low environmentally relevant concentrations, cause the suppression of antioxidant defense systems in strict zebrafish, consistent with signs of oxidative and carbonyl stress and DNA damage. Also, exposures provoked endocrine disruptions, registered as expression of phthalate and depletion of triiodothyronine and neurotoxicity. It might be some kind of antagonistic interaction between chlorpyrifos and Roundup when they are in the same media. Remaining a major challenge in today's world, water pollution has become a top priority for sustainable development. The 2030 Agenda for Sustainable Development brings water quality issues to the forefront of international action. We can assume that runoff from agricultural lands into the surface water containing even background concentrations of organophosphate herbicides may pose a risk to non-target organisms. The introduction of Chlorella in the amount of about 100,000 cells per liter into the environment didn't show a significant bioremediation effect on the harmful effect of studied herbicides for fish, which doesn't exclude the positive impact of algae on the functioning of the ecosystem as a whole and requires further comprehensive research. Introduction of algae in terms of decreasing pesticide toxicity requires more detailed hydrobiological analysis in favor of finding a prospective approach for the removal of pesticides from freshwater bodies as an effective tool for sustainable development and pollution control. Thank you so much for your attention.